The purpose of this presentation is to provide a recent overview of the neuroscience surrounding PTSD and EMDR, which are relevant to Savyn. To start, we're focusing on fear conditioning, as we know that this is a central framework that is being used to understand PTSD. And this occurs where fear can overgeneralize the negative emotions that are associated to stimulus, to people, situation, or context in trauma. And these generalizations can also be perceptual or conceptual. And these have been attempted to be modeled in a lab with fear learning paradigms, which typically involves a unimodal sensory conditioned stimulus that is paired with a unconditioned stimulus, which could be in the form of a foot shock. And this is used to study fear acquisition, generalization, and inhibition. However, this view is simplistic in the sense that, in reality, the stimulus can also form indirect associations with another stimulus without reinforcement, which appears to be uniquely human in the sense of higher order associations, which could be formed through such as second order conditioning, where the first conditioned stimulus becomes paired with a second conditioned stimulus, or it can be in the form of sensory preconditioning, where the association occurs before the actual trauma. So how does this relate to the neuroanatomy surrounding PTSD? Well, there are several brain regions that have been consistently associated with PTSD, including the amygdala complex, the insular cortex, and some areas of the prefrontal cortex, including the subgenual and the dorsal anterior cingulate. And in this circuit, the amygdala acts as a central control that is involved in regulating fear conditioning, with the inputs into the basolateral nucleus that facilitates fear learning, where the sensations of the conditioned stimulus interacts with the aversive information from the unconditioned stimulus, which leads to the consolidation of the threat memory through a long-term potentiation-like enhancement of synaptic efficacy. And what this results in is any future activation of the conditioned stimulus sensory engram alone becomes sufficient to activate the pathways of the unconditioned stimulus. The central regions of the amygdala then sends the output about this fear to other brain regions, including the hypothalamus and the brainstem. So there is this interaction between the medial prefrontal cortex and the hippocampus that both regulates the amygdala outputs to the subcortical regions to elicit these fear and panic responses, including the increase in heart rate mediated through the hypothalamus, the locus coeruleus, the dorsal vagal connections, the increase in respiratory rate through the peribrachial nucleus, the gastrointestinal distress through dorsal vagal nucleus, the increase in startle response through the reticularis pontine cordalis, the freezing and anxiety through the periaqueductal gray and the HPA axis activation through the paraventricular nucleus. And there has been studies that show how neuroanatomy could change in PTSD patients. For example, there appears to be a smaller volume of the hippocampus, which may relate to the modulation of the fear memory responses and the ability to discriminate specific fear cues as opposed to generalization. Another brain region of importance is the subgenual prefrontal cortex that exerts inhibitory controls to threat-related memories, which in PTSD patients, this region appears to be reduced in activation. In addition to the responses mentioned before, they are also known to have sleep disturbances in PTSD, and therefore the pathways surrounding sleep may be relevant to PTSD. For instance, in the study, they were looking at how during rapid eye movement sleep is important for processing emotional memories through the theta frequency inputs into the infralimbic cortex. And this has some relevance to PTSD through the ability of whether this process is able to suppress a potential memory. So this occurs through the interaction between the infralimbic cortex with the amygdala that forms a strong connection from the projections of the infralimbic cortex to the basal nucleus of the amygdala, but the reciprocal projections are weaker, which leads to the ability to suppress the memory. And looking at the overall circuit, there are assemblies that are present in the prelimbic cortex, the infralimbic cortex, and the basal nucleus of the amygdala, which each of them have these cell assemblies, which are a group of cells that form strong interconnections, encompassing this excitatory neurons shown in blue and the inhibitory interneurons shown in red. So in the context of fear memory, let's say this is BA1, which represents a fear memory. It has connections with this region BA3, which is a cell assembly that is related to fear suppression. And you can, from the diagram, you can see that the connection within each cell assembly is 100%, as well as the connections between the assemblies of the same memory. But for all these other connections that are not related to the same memory, they are only 10% connectivity. On top of that, some of these synapses are governed through the spike time-dependent plasticity, which are the connections shown in color, that depends on the pre- and post-synaptic events that occur in these circuits. So how exactly does EMDR play a role in this process in terms of the neurocircuits of PTSD? Well, one of the key frameworks of how EMDR works alludes to the adaptive information processing theory, where usually new experiences would assimilate to existing memory networks. But in the case of PTSD, the memories remain unprocessed, which means that it still contains all of the distressing elements of the memory. And this is where EMDR is able to help. And emerging mechanistic studies have been conducted by scholars like Cyril Herry from INSERM in France, their lab, as well as Hisab Shim's lab in the Institute of Basic Sciences in Korea, where they had mice models with fear conditioning and they simulated EMDR through a eyelid electrical stimulation, where this intervention was found to reduce the rate of freezing in these mice, which indicates the improvement in the fear conditioning response. Furthermore, through optogenetic experiments, these studies were able to elucidate the involvement of the circuit from the superior colliculus to the medial dorsal thalamus, which leads to the inhibition of the neurotransmissions in the basolateral amygdala. And there's also potential relationships of how EMDR is able to modulate the broader fear and threat response, where in the amygdala you can see there are these parallel circuits, but most notably in this diagram is the distinct molecular signatures that are associated with each circuit, like PACAP and TAC2, which can be modulated through EMDR but requires further evidence to support this. In the context of PTSD, there are also changes in anhedonia and ovulation, which are common comorbidities of PTSD, which shown in this diagram is also part of this circuit, shown in blue, the appetitive circuits that leads to these symptoms, and EMDR may also be modulating these circuits. Despite the previous slides possibly appearing to suggest that PTSD may be a disease with a homogeneous outlook, however, it is important to note that PTSD is actually non-unitary with different dysfunctional processes. For instance, there is the complex PTSD, which shows the core symptoms of PTSD in re-experiencing, in avoidance, and the sense of current threat. But in complex PTSD, there are additional dysregulations that have been associated with affect to self-concept and the dysregulation in relationships due to the sustained and multiple exposure to trauma. Another subtype of interest is the dissociative subtype, where the patients exhibit depersonalization and derealization, including in the neurocircuit to exhibit increases in the medial prefrontal cortex modulation of the emotional state. In the dissociative PTSD, they also display opposite brain activation patterns to classical PTSD, including emotion overmodulation as well as dysregulations in the functional network connectivity. There are also increasingly other subtypes of PTSD that could be defined through differences in behavior, in physiology, in blood-based biomarkers, and in voxel-wise brain activation patterns during cognitive tasks. Insofar as the diversity in subtypes of PTSD, there are also correspondingly diverse range of therapies that exists for PTSD. And here I will mention a few recent ones to note. One of the most recent notable study in contemporary therapies for PTSD is through the use of neuromodulation, where an intracranial EEG is implanted into the amygdala, as you can see from the CT scan, and they monitored the PTSD symptoms during the patient's exposure to negative experiences using a emotional image task and script-driven imagery task. And in PTSD patients, when they undertake these tasks, these were associated with increases in the amygdala theta band power. And with the use of closed-loop neuromodulation across several times across the year, these led to the elevations in the low-frequency band power, as well as associated with decreased in PTSD symptoms. In addition to neuromodulation, there are also other contemporary therapies to note that could complement Savyn, and many of which we have also started to work on, including neurofeedback, the use of chatbot for recurrent emotional support, personal assistance, and monitoring. And increasingly, these have started to leverage large language models like ChatGPT. There is also the use of virtual reality during exposure therapy. There's also musical therapy, behavior activation, gamification, which are all important in parallel with the existing pharmacological treatments, which unfortunately many have side effects, does not fix the underlying issue, and have minimal effects on lifestyle impairments. Despite the progress in the understanding and therapies of PTSD, there still exists a lot of challenges, and in part, this is because of the fear conditioning model and clinical research have been ill-defined that leads to this conceptual vagueness that impedes the translational efforts from preclinical models. Therefore, there is cause to improve the constructability of studying PTSD in terms of the mechanism of the disease as well as the trigger. There is also improvements needed for the face validity in the behavior and the biomarkers exhibited in PTSD, as well as improvements in the predictive validity in whether or not the supposed trigger of PTSD does indeed lead to the disease, and how the different therapies may have on the observed effect of the diseases. There are also challenges in the ability to demarcate PTSD from other illnesses in terms of specificity, accuracy, and homogeneity. Therefore, there has been recent efforts to use the research domain of criteria to conceptualize PTSD, which is the use of different neuroscience constructs to try to explain fear from the molecular level all the way to the behavioral level. But unfortunately, this is also limited because conceptual jumps are still required. For example, take a neuroscience construct, valence. There still needs to be this leap of inference when trying to relate that to higher order concepts like fear. Furthermore, the existing measures taken for PTSD may not be sufficient in terms of the readout and the throughput, especially those that are captured qualitatively have not been done sufficiently in PTSD patients to determine how do they differ qualitatively to healthy controls. There is also a dominance in the neurocircuit understanding of PTSD where it overlooks the interactions with other neuroscience systems. So what these suggest is that a lot of advancements are still awaiting to be explored in PTSD, and as these advancements progress, Savyn needs to account for them in further development of our therapies. There are also other topics relevant to PTSD that I think are worth noting and for Savyn to pay attention to, and one of these is the possibility of being able to prevent the onset of PTSD once trauma is experienced. And this hypothesis comes from the animal model work using ketamine and opioid receptor blockers that seem to be able to act as a prophylaxis against PTSD, and therefore suggesting the possibility to be able to prevent the development of PTSD, especially in those trauma that could be predicted and with sufficient lead time that precedes the exposure of the trauma, or possibly just shortly after the trauma exposure, whether some interventions can be made to quickly prevent the onset of PTSD. There is also important to note how it could be different to preclinical models, because humans appears ostensibly have specific ways of regulating different parts of our cognition. For example, in emotion regulation, there is this conscious awareness to the goal that we want to achieve, which in jargon terms is called the explicit process. And at the same time, there is the change process, which is the control process. Both the explicit and the control process requires this representation, interpretation, and comparison. And what this results in is a diverse form of emotional regulation strategies that could range from cognitive reappraisal to emotional suppression to attention distraction to explicit expectancy-based automated processing, which an example of this is the placebo effect. There are also a lot of advancements that have been made in the field of neuroscience which have not yet been applied to PTSD, but could potentially be important for understanding and developing therapies for PTSD that Savyn should keep an eye out for. And part of this involves the conceptual frameworks of different phenomenon. In particular, I gave an example of, in the context of social cognition, which we know that in some PTSD patients they exhibit an impairment in social cognition. So therefore, conceptual frameworks relating to this cognition may be important in terms of how people represent other agents, where there are different theories of belief-based computational modules, such as this one called the depth of mentalization, which models how the self is able to see the beliefs and the intention of the other agent. And this process can also be recursive, where there are inferences that occur throughout the interaction, and it can also be collective when there are multiple agents present. In addition to advancements in conceptual frameworks, there are also advancements to the technical domains that allows a higher spatial coverage and higher spatial resolution during neuroimaging. One example to note is the ability to capture neurons in multiple regions at once, with the development of ultra-fast 2-photon imaging that achieves kilohertz frame rates that captures fast neural activity, as well as positive going voltage sensors called spiky G that have improvements in the sensitivity of spike detection. The brain is also a noisy environment, so there are also advancements in the denoising algorithms like DeepVid, which together combined enables the cutting edge frontier for the ability to neuroimage simultaneously 100 neurons at a single cell resolution for more than 1 hour in awake free behaving mice. Furthermore, from the prevalent genome-wide association studies of PTSD, these suggest the possibility of using gene therapy for treating PTSD, which involves the use of gene editing. And these have been shown to be possible where, in mice models, the viral delivery of the Cas9 protein and the guide RNA to a particular mice was found to affect their memory formation, which has relevance for PTSD. And there are also a lot of advancements that has been made in gene editing, such as prime editing, that doesn't require the double-stranded break or repair templates, but instead uses a fusion protein of Cas9 in the case and the reverse transcriptase enzyme, plus the prime editing guide RNA that can facilitate precise gene edits. And this is important for Savyn to understand a lot of these new therapies that are coming on that requires this complementation to what Savyn is trying to develop, and therefore being able to provide a holistic care to PTSD patients.